I'll start. So great. I'll start by giving some background on electronic health records, or EHR, and then naturally we'll talk about a reproducibility challenge and our related work on a pipeline for processing electronic health records data for real-world evidence. So just so we're all on the same page, an electronic health record, or EHR, is exactly what it sounds like. It's an electronic record of a patient's interactions with the healthcare system. So every time a patient goes to the hospital or sees a physician, all the information that's documented about them is recorded in their EHR. So these are things like diagnoses, the reasons why they were there, as well as any labs or meds, procedures, and then a huge volume of information and clinical notes. And so when we think about using this data for research, the key thing we want to keep in mind is that the data is a byproduct of clinical care. And so to be optimistic, that means that EHR data is really extensive. Like I just showed you, for each patient, we have a longitudinal record, and at each encounter, we have information on numerous fields. And typically within a health system, we'll have hundreds of thousands of patients. And importantly, these are patients that are receiving treatment in real life. So they can be more representative than what you'd have in a controlled setting, like a clinical trial. Unfortunately, in the last decade, there's been increasing EHR adoption worldwide. So of course, the natural question is, can we actually learn anything from this data? And I feel like every time I try to take a break from work, there's some sort of futuristic looking article like this. This is from the Wall Street Journal just a few months ago about the promise of electronic health records to improve the treatment of patients in real time. And what's discussed here is this idea of a green button. So a magical machine learning powered button that a physician could press. They could identify patients that are similar to the one in front of them to help them make a better decision, or perhaps find some unanswered clinical question. But this idea of a green button was actually proposed almost a decade ago in the clinical research area. And we all know that when we see a physician, there isn't a green button that they're interacting with. This is still something that's far in the future. But where we have made a lot of progress is using electronic health record data for research. And today we'll focus in on one popular area, which is real-world evidence, or trying to understand risks or benefits of medical treatments from real-world data, like electronic health records. But I wouldn't be here today if there wasn't a challenge with working with all of this data. And as you might imagine, the data wasn't designed for whatever purpose you'd like to use it for. It's really quintessential observational data. So you could imagine every statistical challenge that you can think of arises. But even before you get to the analysis, the first problem you'll encounter is that EHRs do not have readily available information on phenotypes. And so when I say phenotype, in contrast to the biological literature, I just broadly mean any sort of characteristic about a patient I can infer from their electronic health records. So these are really rudimentary things like, does a patient have a disease? How severe is it? When did it start? How did it progress? Did they respond to treatment? So you can imagine that phenotypes are really foundational for any sort of EHR-based study. We use them to identify populations and also to characterize them. So more concretely, a comparative effectiveness study, for instance, suppose a rheumatologist is trying to understand whether one biologic treatment for rheumatoid arthritis is better than another for reducing symptoms, they would need to do phenotyping. So first, within their electronic health records database, they'd have to figure out who has rheumatoid arthritis. Then the next thing, they need to figure out which patients received treatment and who responded. And so while they seem like really basic things you'd expect to just readily be sitting in your EHR, they're actually quite challenging to infer. And I think it's captured really well by this quote from Dr. Celli from MIT, who's an expert in the field, that just highlights this key point that this is really complex data that really has to be massaged for us to make meaningful use of it. And at a high level, we can understand this by just zooming in on the two flavors of EHR data. And the first is structured data. So these are data elements that are relatively easy to extract. We can query a database to get access to them, but with that simplicity, we lack a lot of context. So just using rheumatoid arthritis as an example again, despite their name, diagnosis codes don't map one-to-one to disease diagnoses. There was a study a few years ago that was done at a large health system in Boston. For instance, they said, what if we just found patients with greater than 3 diagnosis codes, said they had rheumatoid arthritis? Well, it turns out among those patients that they identified, they had physicians review charts, only about half of them truly had the disease. That's because rheumatoid arthritis is really complex to diagnose, so patient can be miscoded many times. There's also just errors in the data. And so fortunately, there's unstructured data, which contains more rich information, but it requires a lot more processing that might help us get at these patient phenotypes. So just to illustrate this, medical text is a whole another field, but it's quite complex to analyze. For instance, the way a physician can write rheumatoid arthritis can be many ways. So fortunately, the National Library of Medicine has created a dictionary of over 1 million what are called medical concepts to map like clinical terms to one umbrella term. But this is just a first layer of processing that needs to be done for any sort of phenotyping problem. And so as you might imagine, phenotyping is really a combination of both the structured and unstructured data. And it's the task of the researcher to aggregate it to extract patient phenotypes. And so you would think that phenotypes being the foundation of EHR-based studies and that they require a complex amount of processing, that the information on how they were defined would be part of how people describe their research. But this recent study found that only 5% of studies release any information on the phenotypes that were used. We looked specifically at phenotyping methods that use machine learning and found that only 1/5 of articles actually released their code or any information about data. And so this has a huge impact on reproducibility of any sort of secondary studies like real-world evidence studies that we'll talk about today that require a lot of information on the complex data processing that's used. So I'll pass it over.
Okay, yeah, thanks, Jesse, for a fantastic introduction, and I will share from my side. Okay, so as we transition from, in general, to use EHR data for clinical research, the focus now is starting to be focusing on the real-world evidence. So in about like 25 years ago, the legislation has already like initiated the process of using real-world evidence for regulatory decision makings, and then FDA has also recently published some guidelines that how they can implement this decision from the Congress. And the one good benefit that we see in using the real-world evidence, especially generated from EHR, is like the study cycle will be much shorter, right? So in a traditional study, we have to plan for the study and then have the recruitment of the patients, participants, and then wait for a few years until we see the outcome, and then we can conclude what's the finding. But for EHR, it's a retrospective process, so the data has been routinely collected by the providers, so that the data collection is much faster, and then we just have to find a best way to extract information from the data, and the study cycle will be pretty short. And it will be very useful due to like trials have limitations in the generalizability outside the very limited eligible patient cohort, and it might not be adapted to very specific subgroups, and then sometimes there's a feasibility concern and or ethical concern for assigning some patient to the control arms, and also there is due to the cost of running a trial, the power sometimes is compromised. So that there is like a lot of opportunity for other a resource of evidence to like supplement that. And some of my colleagues have justified in a summary paper that real-world evidence had a record in supporting regulatory decision. So this is not a new thing, just people didn't invent this word before. And then there is also a trend to use a real-world data to supplement trial recently in this summary. And in the guideline that I mentioned from FDA, they actually call for a basic characteristic for this evidence should be transparent and reproducible, right? It's very basic. And in the past few years, there's some of a team at Harvard have started this process of trying to advocate for pre-registration and pre-specification of the statistical analysis, so that people have minimal, manipulation of the data. Although these work are very important, at least the first step toward transparency and reproducibility, I realized like there is an important missing piece in that process, is how we get the data for the statistical analysis. So first of all, I think the like in their specified analysis plan, the data creation element is missing, as Jesse has just showed in her own summary. And also there is a lack of a step that we can validate the quality of the data, especially in those examples. That team actually focused on the claims data, like we only have those ready to extract structured data, but we don't have any other context we can validate those quality. And just in response to that challenge, recently with Jesse together, we worked up a tutorial for the preparation of EHR data for real-world evidence. And the purpose of the tutorial is to provide the standard data creation protocol so that the future study can include as a part of the design to pre-specify the data creation process. And also we try to summarize all the state-of-the-art computational tools that we use so that we have the best quality, right, from the EHR. And we also provide a validation strategy so that we have a quality assurance, right? Regardless of what initial data quality we have from the EHR from different institutes. And we also provide a guideline how we can prepare data for a robust data causal modeling so that we can adjust for confounding to deliver faithful real-world evidence. And now I will just basically zoom in a little bit to briefly talk about what we do in the folder modules that Jesse has shown on her slides. The first part is the data harmonization. Once we are thinking about we want to run a study using EHR data, we're fundamentally trying to do what the picture is showing. On the left, we have like huge amount of data in all kinds of format, but then we want to squeeze them into a reporting form so that we can analyze them as in a traditional like a baseline characteristic intervention and outcome format, right? So that's actually quite challenging because those information are scattered everywhere, right? So we have to do a few things to make it possible to utilize EHR data. First, we have to organize different data types into a better way. First of all, we need to group those structured codes into, for example, like Jesse showed, like a diagnosis and a medication and procedure and labs. And then we also have to sequence the free text data because that's not easy, for analysis into something we can run. And we also want to have specific tools that can extract those information from language or images. And then more importantly, we also need to map the design from a traditional study to those information where we can get those variables from the EHR. And then for feature extraction, once we map those features into this EHR, we need the specialized tools to get the accurate information about those clinical variables. Depending on what kind of a format the data it is, we have list out some existing tools that can help people to get binary status, time to event, or from text, some specific numerical measurement, or from image, certain diagnosis. And we also the role of these feature creation in the future curation in the data creation is like we will construct how we can use this tool is like we can construct the disease cohort, then we can zoom in to find the intervention arms for the comparison of the treatment, and then we can also identify eligible subset for the study, and then we can curate the endpoint in the follow-up. For the quality assurance, we would propose a validation with partially manual abstraction of data. Basically, we ask a clinical expert to read the medical record and to assess the, give us gold standard labels so that we can assess data quality. We can also calibrate our initial data extraction, and we can build upon these gold standard labels to facilitate a robust statistical analysis. And to model our data to estimate the treatment factor, we have a list of strategies that are also provided in the tutorial that is robust to the data error, right, from like a naive extraction of the data, and also we can account for confounding from different sources. First is we have a way to use a data-driven way to capture the true confounding from high-dimensional summary data of EHR features. And we have also a way to account for the change over standard of care over time, and so that the calendar time can also be a potential confounding. But all of this, we need to have the data that's prepared accordingly, so that though this subsequent downstream analysis can be run with. And for future directions, we need more tools for current available features. For example, the disease activity and the progression, the severity, they are like not routinely collected as also in a very non-structured way. And also some important measures of quality of life, like mobility, cognitive measures, is also difficult to get currently. And also for the baseline characteristics we adjust for confounding, lifestyle and social development health, they have been shown to be very important in many big observation studies, but currently it's not readily available in the framework. And also in future, we might also discuss how we can harmonize across different healthcare systems, as they may have different coding system and then administrative rules. And especially when now we are focusing on comparing the treatment effect over the entire population. So it's like an average treatment effect, but in the future, is something we can zoom in to look at is like how we can identify the person, like basically individual level heterogeneity, and to provide a personalized treatment guideline. So that's my part. Yep.